Welcome to the session B4 on the art of manipulating genes in the area of food and agriculture. Let me start by reading a citation from the program, which I like actually very much. It is a citation from Gulliver's Travel book, and the citation goes as follows. Whoever could make 2 ears of corn or 2 blades of grass grow upon a spot of ground where only one grew before would deserve better of mankind and do more essential service to this country than the whole race of politicians put together. That's what we all try to increase agricultural productivity, because we feel there's an urgent need for this. The subtopic of our discussion are the so-called GMOs, transgenic plants, and you all know that this is a very difficult field in discussions because it's extremely controversial. And in your program it reads further, what are GMOs? There will be no description of what GMOs are. We assume that everybody has an idea what this is. But are they friends or foes? This will be certainly a topic for this discussion here. For some, it is the solution of many of society's most pressing issues. I don't think that any serious scientist believes that GMOs are a silver bullet which solves all problems. But those who are defending this technology have the experience that they can have substantial contributions. For others, it is a catastrophe waiting to happen. And we see in the media that many around the world are, since 25 years, waiting for the catastrophe. And they are very disappointed that the catastrophe didn't appear yet. But they do everything to prevent that this catastrophe may ever appear. So what are the impacts of GMOs on society so far? And what are the expected complications and benefits? We will have 3 speakers. One speaker dropped out, and I'm very grateful that Professor Mark von Montagu was so kind to jump in and speak about the possible benefits of transgenic plants. Eric Huttner, on my right-hand side. Eric Huttner is General Manager since 2001 of Diversity Arrays Technology in Australia, a company delivering high-throughput, low-cost plant genotyping services. So, Eric Huttner represents the most advanced sophistication of breeding, not using GMOs, but genetic knowledge about plants, precise genetic knowledge. His expertise is in plant breeding, so his background is a plant breeder. Genetics and molecular biology, so he understands this side from the basics. He got his PhD from the Institut National Agronomique in Paris, and he had positions at the Chinese Academy of Sciences, Shanghai, of INRA, with the National Research Institute in France, in Versailles, of Limagrain, which is a biotech company, which also has a seed company in Canberra, of BioGemma in Paris, and of Cambia, Australia, which I find specific interesting because Cambia is an attempt to make modern plant development biotechnology available to developing countries free of charge. So to find a way to get around the limits of intellectual property rights and to develop own technology which can be used free by developing countries. Cambia is supported or was supported by Rockefeller Foundation. I don't know whether it's still. So Eric Huttner has a very wide background, and he will speak to us on modern plant breeding by high-density genetic information. And the key topic I picked out from the abstract is that genomic selection, a new breeding strategy where genetic profiles can replace phenotypic selection. So, plant breeders normally work in the field to look for phenotypes to decide which is the most promising one. Now, with the genetic tools we have, you can try to identify interesting genotypes already on the basis of the genetic information. And that will be, I think, the major topic of this presentation.
Yeah.
Please. Yeah.
Good afternoon. So it's a great pleasure to be here in Alexandria again. I've been here before in a few BioVision sessions, and I. Oh. Okay. And I'm really grateful for the organizers to give me this chance again. As Ingo explained, I am going to talk about trying to address the art of manipulating genes, but in a broader context than just GM crops. So the first thing, I'll give you a short introduction about what I think about plant breeding and placing it in the context of what Ismail Serageldin talked about in the first day, the information revolution. And then I'll briefly take one example of GM crop, which a good friend of mine in Australia, Dr. T.J. Higgins, allowed me to use to just illustrate even further what GM can do, and particularly for Africa. And then I'll address the core of my topic, which is the cost-effective, high-density genome profile in crops and what it can do for plant breeding. And I'll discuss these applications and how these technologies and the conjunctions of technologies can actually change the way we do plant breeding. Now, the first thing I want to say is that crop improvement does matter. We've seen this picture in this conference already quite a lot, so I won't go into much detail. And I will acknowledge what we all heard in many sessions, that the solution or solutions to world hunger are not only in science. In fact, they are probably not mostly in science. However, I do believe science has a big role to play, and it is important to keep improving better crops as well as provide the right governance, the right socio-economic mechanism for development, the roads, the infrastructure, and all the other things. But my skills and expertise are deployed in the field of plant improvement. Now, improved plant varieties are made of a combination of genes, and it's important to understand that the value of the variety, the performance that it delivers, are made by the combination of these genes. And it's also important to realize that breeding operates for the benefit of a range of different stakeholders. So the farmer wants yield and he wants profitability, he wants to make money by growing the plant, and also he needs for him, her, and her family to protect the environment so that there is a future. So all those parameters are really critical to address. At the same time, the customer, the consumer of the product wants good quality food at the price that they can afford, and I think we all also need to make it sustainably so we still have an environment to operate in the future. Now, to combine the genes that you need to create a good plant variety, you can use classical breeding, and I'll spend quite some time on it in the later part of the talk, but of course you can use what's been termed genetic modification in many ways. And so I'll quickly go through an example of this, which is not my work, of course. Now, the topic of BioVision this year is science for society, and breeding new variety is really science for society. And I ask you, have you thanked a plant breeder today? Now, the example I'm going to talk about is the transfer of the BT insect resistance gene into cowpea in Africa. My colleague and close friend TJ Higgins is a scientist working for CSIRO, the Australian Public Research Organization. And in conjunction with the African Agricultural Research Foundation, he's transferred the BT gene into cowpea, which is an important food for people in sub-Saharan Africa. Now, the crop is seriously affected by an insect, a lepidopteran insect, Marduka potborer. And when you transfer the gene, you can actually induce insect resistance. So here is the insect, and here is the unprotected on the left and protected on the right leaf of the plant. And so TJ has worked for several years obtaining the best plant for this phenotype. And the field trials for the plants are now starting in several countries of sub-Saharan Africa, and they do suffer partly from the same limitation which Ingo Potricus this morning addressed, in the sense of the weight of regulation is making the development of such product into a practical product for the farmer very difficult. Now, I'll go back to plant breeding. We were talking about integration, combining a large number of genes. Now, over the past 30, 40 years, people have tried to do this task by using, in addition to the traditional skills of the breeder in the field, using molecular genetic marker, that is DNA marker. And those markers have been useful for a range of tasks. To study germplasm diversity, identify the diversity in the collections, to identify and possibly track some useful genomic regions, and to follow some particular traits in routine breeding. However, there hasn't been a massive use of those technology for several reasons. The cost of developing them is quite large, and for a long time, there was no economic advantage for the breeder in going for DNA markers rather than proper phenotypic observation. This was particularly true for species for which the economic value is not very large. And so although breeders of crop of the industrialized world were investing in molecular marker and using them for many species in the south, it was just not practical. And so 10 years ago, my friend and colleague Andrzej Kilian asked, can we do better for orphan or small crops, as well as getting more marker to be able to deal with complex traits and complex germplasm, including highly polyploid plants. Now, when you have molecular markers, if you're dealing with simple traits, there's a lot that you can do. For example, with disease resistance gene, one gene has usually a very large effect. If you have a marker for the gene or a marker that's closely located close to the gene, you can follow the resistance, and that's an alternative to doing the phenotyping. With medium density profile, you can use the profile to transfer regions of the genome. But for complex trait, you need a lot more profile because you need to combine a lot more genes. And as I was trying to explain, until recently, it was actually not very practical because not really cost effective. At the same time, the increase in data density requires new information technology and infrastructure and software to deal with the data. And all those things, the technical aspect of producing the data as well as analyzing the data, came about to become reality in the very first years of the 21st century. So I'm going to take the example of the technology that the company I work for is actually using. Of course, there are other technologies, but we found out from 2001 when we started to now that there are actually not many alternatives which deliver the actual quality of the marker system that we are using. This is changing with changes of technology, but for the decade of 2000 to 2010, there was actually not many alternatives doing what we did. The original method was invented by Andrzej Kilian, who is the young man on the right, and he's talking here to Norman Borlaug on the left. And Andrzej invented a method which is based on the reduction of the complexity of the genome that you want to analyze. We produce what we call a genomic representation, and in a minute I'll show you how it works at the molecular level. And this genomic representation represents between 1% and 0.1% of the genome to be analyzed. And this genomic representation, this highly reduced description of the genome you analyze, can be analyzed with two platforms. We deployed a microarray platform in the last 10 years, and in the last year we've started to implement a DNA sequencing platform. Now, how it works is that what we call a marker is a genomic fragment, the presence or the quantity of which is polymorphic in a genomic representation. So if you have two plants here, and you prepare the genomic representation here, these genomic representations have fragments in common. They are in black. They also have fragments which are different from plants to plant. They are in color. And then they have some sequence-based polymorphism which are represented here, which are called single nucleotide polymorphism. Now, on the microarray platform, you just have to hybridize the genomic representation, and some of the spots on the microarray will light up or not and detect the polymorphism. With the sequencing platform, you just take the genomic representation, use one of the recently developed equipment which they are sequencing at high speed and low cost, but producing short reads of data. And you then can count, you can then count the presence of each fragment, which give you a presence-absence polymorphism, or you detect the SNPs by the sequence. So these are the two implementations of the marker system. Now, once this technology was invented, because we were interested in actually making it work for people, not just having invented something, we established a DNA to data service, which we operate in Canberra, Australia, to this day. So the people who want the marker data, they send us the DNA, and we report to them the result. These slides illustrate that we have on the microarray platform a range of density of markers. At the top, we show a high density array where we assayed between 7,000 and 15,000 markers in one assay. And that's very useful for doing building genetic maps, doing QTL mapping, association mapping in various populations, and we'll come back to it towards the end. And the cost of doing that to our customer is around US cent per marker analyzed. So if you send us a plate of 100 samples of wheat, we can analyze it at high density for about $5,000 to $7,000. And what we process is DNA. You extract the DNA, we process the DNA. We cannot import plant material in Australia because of quarantine concerns. We also have developed a low-density array, which is illustrated at the bottom of the slide, where we only assay between 200 and 500 markers. And with this density, you can do a range of applications, for example, guaranteeing seed purity and doing some genetic identification if there's a question what variety is a particular plant. And at that density, you can also do genomic selection in some context, but I'll come back to that. Now, that's just an example of what people receive. We send them a scoring table which contains the list of marker polymorphic in their material, the scores, and some parameters which reflect the quality of the marker scoring. And that's an important thing compared to other marker systems, is that we have a quality parameter that allow people to filter the marker according to their needs. So if they are happy to tolerate a higher error rate, they can take marker of lower quality. If it's absolutely critical that there are no errors in the data set, every marker system has an error rate, then they take marker with higher quality. Now, when we do genotyping by sequencing, when we don't use the microarray platform, but the sequencing platform, we, as I explained, sequence the genomic representation. For each sample, we generate millions of tags, little sequence, about 70 base long. And for each of the fragments in the genomic representation, we aim to have between 100 and 1,000 tags. And so we report the score, 0 or 1, for each plant, if a tag is present or absent, and that's a specific subset of polymorphism. And for the other polymorphism, we report to the people the sequence that we observe in comparison to a reference. So for example, if you have SNPs in a particular marker, we report the score, the plant for which the allele is the reference allele with a 1, and we report the plant for which the allele is the SNP allele, the polymorphism, with a 1. And you can see occasionally that you have heterozygous plants which are 1 for both alleles. And we have some quality parameters as well. Now, the service is available at the last I looked. We have a very large database to keep track of that, and I don't know those things by heart anymore. But I think we have 72 crop plants and relatives of crop plants for which the technology is available off the shelf today. And we can develop the technology for any new crop in a matter of a few weeks if we have the interest of someone. By the way, I realize not everyone works on plant. We are also analyzing a metagenomic sample of microbes, and we are starting with the sequencer to analyze animal sample, but our key interest is in plant. Now, with the marker profile that I presented, you can do variety fingerprinting and diversity analysis, and that's usually one of the first things people should do if they do plant breeding. They should have an understanding of what the diversity is in the material they are going to manipulate. It is important to recognize, for example, duplicate sample. If you have large collection, you don't want to waste your time propagating things which are copies. You also want to design your crosses and your crossing schemes in agreement with the diversity variation that you observe. So this is usually one of the first things people would want to do. We create linkage maps, and so the population can be mapped. Someone is giving me a telephone, thank you. Especially working in Egypt, mine doesn't, so that's good. The good thing is that you don't need to prepare anything special beyond the data set. You map as you make the cross. And this profile can be used to introgress material from exotic germplasm. And a limit example of this case is a GM plant, which you need to transfer into local varieties. We also do bulk segregant analysis. And once a marker is identified, you can apply it on its own if you want to. I am late? No. I
had 20 minutes. So
but started at 22?
No, we started at 5 past.
Okay, all right. So we have 1.5 hours. Okay, well, yesterday I was told 30 minutes. Okay. Yep. All right, there's a lot more that you can do with the whole genome profile. And the key issue for us is, will this enable new breeding methodologies? In particular, a bolder use of genetic diversity. Can you go in the germplasm deeper and further to get those exotic traits that you need to get your germplasm improved? We developed the software to actually do those tasks, and in the interest of time, I won't go into any detail, but the approaches that are called data mining are particularly appropriate for this sort of task because of the size of the data sets. Now, marker-assisted selection, as I've described, has some limitation, in particular because for many traits, a large number of genes each contribute very little. And so there is no such thing for many traits as the marker for the trait. And this was highlighted in human heredity, in human inheritance by several papers now. For example, on human height, which is a trait which is highly heritable. Despite all the genomics resources and all the SNPs available, people cannot explain the additive variance with the markers because each genomic region contributes very little, but together they contribute everything. And that's where genomic selection comes in. It was first developed for animal breeding, and it works like that. You take a training population, which you phenotype very carefully, measure yield, property in a range of environment, and do it as accurately as you can, because this is going to be the key to your effort. You genotype it using one of the methods that I presented, and then, using a range of statistics, you use all the marker data to estimate the effect of all markers on the trait without any marker selection. And this allows you to calculate a breeding value, and this breeding value can be based on any trait of interest, the yield, the product quality, disease resistance. When you have calculated that breeding value, you take a validation population which you haven't processed that way, and you apply your model and confirm that the model identifies properly the associations. And this gives you a mechanism to use the breeding value in the future, instead of the phenotype, to select the best individual. Now, the feature of this approach is that it's unbiased. You don't need to understand much. You use all marker data. You don't need a genetic map. You don't need to find QTL. You don't need to understand the underlying biology. In fact, it may be extremely difficult, if not impossible, to fully understand the underlying biology because of gene by environment interaction, for example. And why is this coming up now? It's because it's become practical now because we have whole genome molecular profile that breeders can afford, which is only in the last 10 years. And also we have the IT tools, the software and the computers to do it. Now, the model of this, the mathematical model, show that by doing this you can shorten your selection cycle very dramatically, and you can increase your selection efficiency. There's a range of practical consideration which maybe I'll cover just at the end. Now the question is, does it work? And so we're starting to get example with our collaborators and friends from all around the world, and I've quoted 3 publications in the talk, so you can refer to them. And I am not a mathematician, so I can't comment on the actual mathematics behind it. But Dario Grattapaglia at Embrapa in Brazil has asked us to genotype 2 very large populations of eucalyptus, and by using about 3,000 to 3,500 markers, he managed to calculate breeding value accurately for a range of wood quality trait and speed of growth and yield. And so the use of those breeding value allowed to reduce the breeding cycle, and for a perennial crop like eucalypt, it is an extraordinary improvement if you can reduce the breeding cycle even just by 1. At CIMMYT in Mexico, we had a team of CIMMYT ask us to genotype a large number of what's called historical lines, which were genotyped by us and phenotyped carefully. And they again observed a good correlation. And our colleague at Cornell University likewise used a different system based on biparental population. I'm finishing. So the data show that genomic selection can be used in plant breeding. And I want to conclude by saying, in summary, what I tried to say in the talk. First, I want to emphasize that GM technology can serve society because we are addressing safety issue. And I am getting very concerned that the concerns in Europe are slowing the progress in Africa, that people who have no stake are telling other people what they should do. I've shown you that new breeding methods are possible because we have new instruments and new computers to do the work. And thanks to this sort of progress, information becomes knowledge, and knowledge becomes new product. And remember to hug a plant breeder tomorrow. This is our team, and these are the range of people who have helped us along the way. Thank you very much.
Let me explain the discrepancy between our expectations. We have a total of 90 minutes, and we have 3 speakers, which is 30 minutes per speaker without any introduction, any discussion. So that's why I intervened at 20 minutes. Dr. Huttner is innocent because yesterday evening the expectation was that we would have only 2 speakers, but it changed this morning, and I'm very happy that we have 3 speakers to represent the breadth of the area. Excuse me? Yeah, that's good. But this doesn't allow now for questions. We have to go on to the next speaker, which is Konstantin Canavas. He is Professor for Technology and Risk Assessment at the Technical High School in Hamburg. He got his PhD from University of Stuttgart, and he had positions at Düsseldorf, Stuttgart, and Hamburg, and somehow in Athens as well. I'm not quite sure. To give you an indication on what is expecting you, I tried to fish out one sentence from the abstract, which reads the following. The main goal of the present study is to demonstrate by means of real examples how this dependence, meant is from imported GMO products, can amplify even small disturbances in the globalized biosafety network, and how such disturbances can cause economic disasters, in some cases irreversible environmental degradation. So the topic is here, the topic on what do we do with risks, can we manage them, do we have to try to prevent them, and I'm looking forward to the examples. Can we agree on, if possible, 20 minutes that we have time for discussion?
Yes, you stay up at 15 minutes to warn me, okay? Thank you. Thank you, Mr. Chairman. It is pity to have so impressive scientific technological achievements as those presented during the conference, and at the same time so many conflicts on the interface between technoscience on one side and politics society on the other. These conflicts are often on the background, or are carried out on the background of uncertainties in scientific knowledge, uncertainties in assessing the impact of technological scientific developments and in legitimizing political decisions. And it is precisely this interface that my presentation focuses upon. My perspective is that of technology assessment, not of biotechnology, not of genetic engineering. So the debates on genetically modified plants, especially for food use, are characterized by controversial assessment representations. One repeatedly used argument to legitimize the introduction of genetically modified plants for food use, especially in developing countries, is, and this has been stated several times during this conference, is the claim according to which developing countries need genetically modified crops to feed their populations. The counter-argument claims that food shortage results from unequal distribution of existing crops, a distortion which results from the unequal possibilities in the dominant production and distribution system. Such fundamental debates keep under their shadow issues concerning the dependence of a national economy upon controversial resources, that is, upon products like the genetically modified plants and technologies assessed controversially by transnational consumers. This dependence determines the degree of vulnerability of a given national economy in respect with disturbances in the production and distribution of such novel products, which exclusively depend on new biotechnologies, for instance, genetic engineering. In the case of developing countries, the dependence upon imported or exported patented genetically modified plants or genetically modified plant derivatives results into higher vulnerability levels. Now, the main goal, as the chairman said, of the present study is to demonstrate by means of historical cases how this dependence can amplify even small disturbances in the globalized biosafety network, and eventually how such disturbances can cause economical disasters and, in some cases, irreversible environmental degradation. The examples refer to major conflicts related to product transfer among the USA, Japan, and European countries. But the pending question is, what would happen if, for instance, these genetically modified products from which I am going to talk about were produced in a country in the development world, let's say in Egypt? Thank you. So that these will be the three cases I'm going to talk about. The LL601 rice, the Monsanto maize 810, and the genetically modified potato Amflora of BSF. I come to my first example. In January 2006, an exporter customer finds contamination through unknown genetically modified rice in conventional rice stored for export. This had as a consequence a disturbance in the commercial chain. The load was impeded, would not be treated. It was intended to go to Europe, and now it was found, but it was just by chance and good luck that, and that was in June 2006, that the genetically modified rice, the contamination was the genetically modified rice LL601, which was not authorized for commercial use. It should be stated that in the years between 1998 and 2001, experimental cultivation in the USA were carried out on genetically modified rice LL601 of Bayer crop operation, resistant to insecticides of the Liberty Link family. But 2001 stopped and no authorization afterwards. So how this rice entered into the commercial load 5 years later. Actually, this detective story is not important for what happened, because what happened was an economical conflict and not a conflict among scientists. The explanation given, it can be short, was that the years after the experimental cultivation in the USA, the experiments were carried farther, this time by a university unit in Puerto Rico, and but not with authorization to become a commercial free land experiment. So the data who were by Bayer Crop Science were just by chance, because the company was not it was not compulsory that the company held the data. It was just the goodwill, and it was just the chance that she gathered the old data so that the contamination could be easily identified. At that time, identification of an unknown contamination was not so easy. Well, it's not easy today, but it's not so difficult today. So, but the problem of that time was what happens now. What happened? August, September 2006, a chain of reactions. First, surprisingly delayed, or not surprisingly delayed information from the USA towards the European countries and the other countries affected, let's say Japan. Import ban in Japan, import ban in European Union. Not only of this rice load, but there were more rice loads banned at that time. Accusations from the rice cultivations in the USA claiming their rights, the damage by the one case which they didn't feel themselves responsible, and they, from their point of view, they were not. Now, even worse, the genetically modified rice was traced in European markets and magazines. That seems that this kind of contamination was already on the road when it was discovered. Now, the scientific position. The European Food Safety Association declares that there are no toxic hazards, so the problem is no problem from the scientific point of view. This is the risk assessment of the agency commissioned by the European Union declares unguilty. That has absolutely no sense for the commerce, since the rice loads were sent back to the USA not because they're dangerous, but because the commercial chains cannot sell them. So the rice goes back from the European Union and from Japan. There's a price fall to export temporary in the rice in the USA because of this disturbance, or it could, from the point of view of a single farmer, it could be more than a disturbance. Now, lessons for implemented biosafety. In that case, we have first to state that we have deficiency in respect with the traceability and the quality control, and the potential of restrictions considering the irreversible ecological impact. As far as risk assessment is concerned, I mentioned the discrepancy between the scientifically based statement, no toxic hazards. That means, and the socially based perception that when you do not, as a customer, when you do not expect genetic modified rice, you don't want to find it. And if you find it, there is something wrong. And you don't care about minimum and numbers and whatever may be, or the toxic or the toxical innocence of the product. Now I came, just to summarize, what could that mean if it was not the American farmers touched, but if there were farmers in a developing country with no insurances and no the ways of buffering this kind of damage, who was not predicted by the company who sell the rice. My second example is a more bizarre one, because it is not an accident, if you like, that is programmed to happen, but it is something that has to do with upgrade of regulations. It concerns the famous maize Monsanto 810, who produces natural insecticide, authorized for commercial use in the European Union, and confined myself in the European Union in 1998. Now, there are new conditions, 2003, in the European Union concerning the authorization of cultivation for commercial use. That is, and now the point was to adapt a posteriori the authorization conditions for issued authorizations, like in the case of Monsanto 810. Now, this product was not so, it's not a so long perspective from the point of view of Monsanto. Apparently, so it seemed less probable that the company would comply with this, not because they didn't want, but because they were not so eager about this product after so many years of use. And now we have a banning chain banning concerning freeland cultivation of Monsanto 88 because of the change of the conditions in several countries. Well, Austria had already banned on the base of the safeguard clause in the European guideline, who gives the right to countries to deviate from the recommendations of the European of the committee. So Austria was not at that time, but Greece in 2005, Hungary 2005, Germany 2007, France in January 2008, followed on the base of the safeguard clauses. So we get a very diversified landscape of not of regulation, of applying the possibilities of the regulations in the European Union in, say, one political and economical landscape where you're supposed to have a unified regulation, but with clauses which enable different compartments and behaviors. And it is also important to see in this map, at that time, the difference in the application. So you see in France, these production rates at the hectares in the year 2006, 2007, much less in Germany. But what happened is that in Spain, which is not touched by this, it doesn't react in this case, we have the main loads. So from the point of view of Monsanto, this is the actor and not that one. So from the point of view of a farmer, because that's my point of view, you cannot predict what will happen and why it does happen like that. It has not a scientific background, and it has not the background of saving the world from hunger. So how to define national biosafety? You have two levels, or we have two levels, and on one level is the national decision, which is necessary, but we have also the local decision maker, the local level, who goes beyond the horizon of toxicology and also beyond the horizon of the producer of the technology. I come to my last example, which is more, let's say, on the level of the decision of a single company. It has to do with the genetically modified potato type, the Amflora of BASF. It is modified concerning the starch content. Now, this type has nothing to do with food, superficially, because it is intended to have industrial use. The problem is that, and the problem was during 13 years of conflicts with authorization authorities in European Union, that you cannot exclude that this potato enters the animal feed chain. In March 2010, there was an approval of commercial cultivation of Amflora. Some months ago, January 2012, the company, the BASF, stops further development of these, not only of this, but of further GM plants in Europe. And now I quote not Greenpeace, but I quote BASF. No perspective for successful commercialization of GM plants in Europe in the next years. This is not marketing strategy for GM plants, but how to predict it. Now, the explanation that was given is that under the conditions, and one of these conditions mentioned is the comments of the German Federal Constitutional Court of November 2010, which underlined that green biotechnology involving genetically modified techniques is a high-risk technology. So quite in opposition to what we've been heard from several speakers today. Now, the German Federal Constitutional Court did not invent anything, did not change any law. The law existed since 2008, and the Constitutional Court just commented and underlined already existing regulations who were known and who didn't touch anybody, at least not at BASF at that time. So just imagine that you have farmers in a country with no networking of insurance, etc., and who depend on one product of that kind, and then you have a decision like that of BASF. We stop we stop research in Europe. We continue the research in the USA or in India or whatever, but the market of Europe is not interesting for us anymore. And then you have, as I said, the farmers in another country, in a developing country, depending on this product, exporting some of this, not exporting the whole, but some of this export to the European Union in this changed landscape. So the problem is, from my point of view, the unequal distribution of expected benefit from the, cultivating the genetically modified plants, and what is the sense of accepting the hypothetical or verified risks. And as I tried to point out in the three examples, there is not a general, an ideological position, but it's just the specific vulnerability of some societies, and I was referring explicitly not to European society, not to the American society, not to Japanese society. I was referring to developing countries who could or would adopt the planting this kind of plants, not only to feed their populations, but also to export a part of this production. My proposal would be not to stop doing that, but my proposal is the informed participation. That is, the ones who are going to get involved in such production should be integrated in procedures of participatory risk assessment to know their customers and not only their producers. And the international protocol, that's my last sentence. The international protocols, who could be for some an ideology or could be an empty word, they can mean something. They are not remedy for everything, but they are symbolic signs of continuous reference for decentralized procedures of control and decision making with a wider societal participation. Thank you for your attention.
Thank you for your presentation. We have time for discussions, and I think the topic is interesting enough to provoke a lot of discussion. I regret that I'm chairman and cannot contribute. So please, can we have light for the table here?
Okay, it's me, Ali Muhammad Ali. I am a chemist in the medical authority here in Egypt. I graduated from chemistry microbiology department. And I would thank all of you, the scientists all over the world, for your efforts. Okay, and I would thank Mr. Eric and Mr. Canavas for their amazing presentation. And let me ask my question. From the point of view, it's me, Ali Muhammad Ali, participating here in this event to know how the world think and move around us. From this point of view, is there any pathway in the field of science encourage fixing our problems that humankind makes, then make a fund for these programs which work on the genetic modifying plants, which I think it has many problems and not 100% safe. And I want Mr. Eric and Mr. Canavas to respond to this question. Okay, thanks.
From my perspective, that is not the perspective of the insider in biology and technology. It is important for the producers of technology, for the distributors of technology, to take seriously the concerns of the layman consumers who do not want to go through a crash course of biology and risk assessment before before buying a crop and before planting a crop. They want to plant, they are concerned with the ground and not concerned with the inside of the molecule of whatever the biology or the biotechnology produces. So it is socially, it is a communication challenge, and that was what I tried to express by the informed participation.
Thank you. May I ask the other questionnaires to try to ask a short question, not give an entire curriculum and lots of explanations. Please.
Thank you. My name is Ahmed from the National Organization of Drugs Control. I would like just to transfer our fears as Egyptians, as developing countries, as third world countries without no authorities. I would like to transfer just the fears that we are afraid of being like experimental human animals for those good for those GMBs, for those brilliant works that could help the world, but we are afraid of being like human experimental animals, Doctor. Please refute this fears.
This was not a question to Dr. Canavas, but this was a comment to the podium.
Please, all doctors, please.
I know that the notion that innocent, poor people in developing countries are kept as guinea pigs for rich European countries. This is just stupid, because all material which is made available so far on the market, either in industrialized countries or anywhere else in the world, has been studied so carefully, and the regulatory authorities have looked at it with extreme precaution, that you can have a guarantee that consuming transgenic food on the market is of no risk to you or anybody else. This has been really guaranteed. And the examples which we have just heard have not indicated any inconsistency with safety or hazard, but it has demonstrated the hysteria of the present situation. And specifically, the ruling of the German court on this case demonstrates brilliantly the stupidity of the lawyers. So I'm sorry. It has nothing to do with any kind of risk, neither to consumer nor to the environment. It's a political move. Yes, please.
Yes. About the fear of being the guinea pig, I just want to say one thing, is that the technology has been tested by the American customers since 1996. So the guinea pigs have been identified, they've been used. There's about 300 or 400 million people in the rich world who have used the technology for themselves, and it is about 15 years later that it's proposed that it maybe would come to your country.
May I,
Okay.
may I just
Please,
one
yeah.
sentence? I think that you're mentioning a political issue, and the reaction, whether it is yes or no, will be a political one. It is not a discussion on scientific terms. That means that every society has the right to say, no, we don't want this product, not because it is dangerous. You don't have to justify on the basis of health. You may say, we don't have that. There is a reaction to this reaction in a situation with another country of Africa, where the one who wanted to give this product said, beggars have no choice. If that is the reaction, then you can comment it yourself.
Please.
I can talk loud, and I think it's necessary to say here a few things which are not so diplomatic. I have my reservation that
microphone is working.
yes, that Professor Canavas is speaking here on a matter where he has 0 publications on scientific peer-reviewed papers. He cannot show he is an expert in this field. I have checked it out in all websites, in the Web of Science. You have 0 publications on the subject. And you have also demonstrated in your talk that you are an absolute layperson and you speak, to say it undiplomatically, absolute nonsense. Because if you want to make a political case, I think you should see that in Europe we have a political system which you first should read about and test what you say and not talk this kind of nonsense here at a BioVision. And I feel shocked and sorry that a speaker like you, as a layperson, can come with his personal opinion, not being based on any science, not social science, no nothing, and no publications, and to give his nonsense here. Sorry to say that. I don't want to go into details. I have about a dozen details where I can prove you wrong with your statements.
Well, I think I should interfere as chairman. This was really tough acquisition, and you should have the right to say something at least in your defense.
No, I think the proposal was to discuss afterwards.
Well,
Okay.
if you don't have any, can you
Can you
say any publications
No,
you made or
please,
not?
please stop.
Yes, it seems to me from what Dr. Canavas says, and what he says is forbidding, but obviously represents the truth in Germany anyway, that GM foods has moved into an Alice in Wonderland world, if people here have read Alice in Wonderland. I really want to ask him a question. If the German federal court were to decide, quite justifiably, that horse riding was a dangerous form of transport, would that justify the banning of keeping horses in Germany?
I mentioned the German court not as a lawyer, but because the quotation is mentioned in conjunction with the decision of BASF. So I don't represent the German court. I don't want to justify the German Constitutional Court. I found astonishing that the repetition of an already existing sentence from law concerning genetically modified foods, just the repetition, the underlining, became an argument, and I'm only quoting and discussing the discourse, became an argument for justifying the decision of a company. I am not the company. I found it absurd that companies just discover something that already existed, was not annoying 2 years, and it became annoying then. That means, for me, it is just superficial. I think that everybody can understand that it is not earnest, but it is just a discourse. And I was quoting the discourse and not science.
Okay. Before we have the next contributions, let me make a quick remark on my comments about the German court. If the German court has stated that GMO technology is a high-risk technology, then they are wrong according to any scientific standard. There are hundreds and thousands of careful publications which demonstrate that this is not true. And it is even your own experience, because there is no documented harm from any use of any transgenic organism anywhere since 25 years. So not only science, but your own experience tell you that the members of the German court must be wrong. And the decision of BASF to move out of Europe was a political decision because they realized that this technology has, on this background, which was demonstrated by the German court, no future in Europe. It was not a decision based on possible risk. Let me focus on two more contributions and then we move to the next speaker.
Dr. Away from politics, FDA banned the importing GMO food, but okay, you can use it for feeding your population of the developing countries, in a sense that they're saying it causes no toxicity. But as a scientific rule, we have something saying the GMO, if it's not safe because it causes mutation, so we have two types of mutation, point mutation and gross mutation. It doesn't cause gross mutation, and this is more, I can say, safer, so it will not kill the person, but it causes point mutation, and it can be inherited to the late generation. So if it doesn't kill the person, it doesn't cause toxicity, it doesn't cause gross mutation, but it's banned. So in what sense?
Okay, I think your contribution illustrates again the hysteria of the situation, because if you are concerned about mutations, you should be concerned about mutations which are real. You may know that plant breeding has a long tradition of mutation breeding, and plant breeders have used every possible chemical and radiation device to induce mutations. And I'm coming from an institute where breeders used a cobalt bomb which was firing gamma rays onto wheat fields for decades and used this material for breeding. And this is not regulated. You are eating it without knowing that this is a result from mutation breeding. And the famous Italian pasta is all based on this type of material. So here you should be concerned. This genetic engineering situation is totally different because everything has been studied in detail to the last molecular level, and everything what is not safe has been excluded. So there's quite a difference between mutations.
In what sense it's not safe? This is the question.
It is at least as safe as everything else you are eating.
Just two comments. One, I think the perception I'm getting is I think a lot of us have to really go back to basic sciences and really spend a lot of time in understanding what the basic science is about. If you don't understand basic science, it's going to cause hysteria, as the chairperson has remarked. The second question is really, second comment is actually to address young Muhammad's remarks that he made. In his remark, he mentioned that until you prove 100% safe, why should you be used as guinea pigs? You please point out one technology anywhere in the world that is 100% safe. That's the,
okay.
I mean, somebody said there is no zero risk technology anywhere in the world. Anything that we do every day, there is something attached to it. So please let's be a little bit sensible about that. Thank you.
Okay, thank you for this comment. Now let's move on to the next speaker. I am sure you all know Mark von Montagu. He does not need an introduction, but still let me give you a short introduction. Mark deserves the Nobel Prize for his work since long, and I hope I'm still alive when he will finally receive it. Thanks, Mark.
I try to stay alive also. Okay, I didn't know what the session was about. The title was Manipulating Genes, so I thought we would talk science. But since the atmosphere that has been created in the former talk, I think first we'll go into there, and then if some time is left, And if you are interested, we can talk science, because that's what's the most stimulating part. But I think I was very grateful for the talk that we have heard, because it underlines the tensions and the wording that is used in communicating. I would not agree with Klaus that if our colleague is not a specialist in plants, that it matters. His points that he brought was with the three cases that are very important cases that happen, very important cases of how society reacts absurdly, that it influences the outcome. If go from the from the Amphora decision, and if a German court says that it is risky, it's a company like BASF cannot go on, because they are not the machines of the planet. They are there as a company to survive. And we know, since people have coined the terminology contamination, that is self-decided. It doesn't exist generally in the food industry, zero contamination. And that has always been accepted. Maybe people don't know that for the flour for bread making, you may have a certain fraction of crushed mice and cockroaches in it. It's just a threshold. And that's all over the world, and that's for all food products. Zero doesn't exist. And if now people get away with the concept that zero should exist, it's absolute nonsense. But society has to learn to live with absolute nonsense, how to react. And you cannot shut up, and that's why we are here, for not shutting up and saying that it is criminal to say, that people would say we want, it's contamination if our methods are so good that we can trace DNA for the transgenic organism there. So that's indeed a case that should be handled much clearer and much faster and more efficiently. And the same with the story of the rice. That was a contamination that happened, that everybody knows is a no meaning, but because in the law there is not written that you can have this percentage of contamination and this rice was not approved, you can call it contamination, and it has cost the organization buyer $100 million. And then, of course, it blocks the whole GM technology, and only what people in the newspapers remember is that there was such an enormous amount spent because they were sloppy and it is dangerous, these GMOs. And that's why I think this talk was very important, that we remember these facts, that we just don't ignore him, and that's a major problem of society. But I don't think it's very constructive to go further in detail. We can do it also in the discussion. I thought that manipulating genes, you wanted to hear what's the latest in genetic engineering and where is the science going. And like always in science, it's very interesting how many new things have come up since in the late 70s we started this agrobacterium technology. And unfortunately, many groups forgot to work on agrobacterium, but it's fascinating how this bacteria makes a certain quorum, this quorum sensing, that you have a film around your plant cell, and that at that moment genes are induced in the bacteria that this transfer can happen. And then how this transfer really happens, the details are not known. It's of course difficult to follow a single molecule, but it's surely interesting molecular biology. But what we have to know, and that what you want to know if you do genetic engineering with agrobacterium, that you need to have hundreds and hundreds of transformants, and that you have to have efficient transformation, and that's why we cannot without the marker genes, because banning marker genes in transformation makes that most of the crops that are in the wild can not be used for transformation because the efficiency for transformation by agrobacterium is extremely low. It's interesting to see why and what happens. But we need absolutely marker genes, and it was very unwise to say, yes, we can do it in other ways. We can do just screening. Then it's giving an advantage to the multinationals who can have laboratories where hundreds and hundreds of people are working, of maybe to Chinese laboratories. Then they can find the efficient and the good transformants. It's just blocking many of the developing countries to participate in this science if you don't accept the marker genes for the transformation. Because, as I said, there are many additional mutations that come up if you do it through cell culture. In the old days, one called it somaclonal variation. Now, with some sequencing techniques of the methylomes, we will understand that it's maybe epigenetic variation that go on. But all the people who have worked in the lab know that you can have quite some good transformants as a callus, but they never regenerate as a plant. And if you do regeneration, it's the untransformed. In the old terminology was a declone. Of course, they have never been cloned, it's just a selection that you did. So there is interesting science at that level. It's for specialists, it seems, but you have to know it and study it. And at the moment, I think it's also worthwhile to know that it is possible in a group to have sometimes sets of 8, 9, or 10 genes that are transformed, that you can regulate the expression of the genes that you will transform. There are systems of vectors. We ourselves developed and improved the gateway vectors. So if you write, people who need some vectors, these gateway vectors that have are available can overcome quite some situations, but still a lot of work has to be done on that. The whole work, we have to realize, and I stressed it this morning, if you want to make transgenic plants, what's most important is the plant with which you start. And with the technology, surely, finally, we will be able to use the best and the high-yielding varieties, but and the varieties that have the properties that you want, but at that moment, you will have to do transformation in so many varieties. And there again, the legislation that has been built up blocks us completely. Because if you, for bringing a transformant to the market, it will cost you 50 million euros, 100 million euros even, because that's the price that some of the major companies say that it costs to bring. Then you have one transformant, and then you will again just cross it with other varieties. But you need to make the very because varieties are so different already. Plants that have been used for breeding and optimal breeding are already very different from the wild varieties. So if you have a tomato or a strawberry variety with a very good flavor, you cannot start crossing it with the transgenic variety that would help you for some disease control. No, you should do it again. So we have to work on all possible varieties with the marker genes, and we have to fight that it is considered as identical, because there is not the slightest reason that it would be more dangerous to start over again in the new variety, because never any danger came up till now. So that's a point that we have to study. A completely other aspect that is, till now we only looked at the plant. There is so much to learn on what this morning I called the holoenzyme, all these microorganisms that grow together with the plant and that influence and that give signals. Recently, it has been seen that quite some microorganisms can make regulatory microRNAs that are important in eukaryotic cells. So the probably there is a lot of signaling that goes on that we till now we didn't know. But anyway, there's nothing to see with danger because that's going on for many thousand years. All of classical agriculture is together with this microorganism and this signaling that's going on. I only pointed out that for the future we will learn so many more changes that we can trigger. We will probably, with microRNAs, be able to trigger some of the epigenetic changes for the unraveling of the chromatin and modification of the chromatin temporary that mimics then some stress situations or that makes that the plant can react and that we can analyze that. So it's very interesting. We'll need more knowledge on hybrid vigor. I touched it this morning. All the seed companies work only on plants that are hybrid plants because they, at that moment, the farmers have to buy the seeds again from this company. But they have an enormous advantage because the yield of the hybrid plants are so much higher. So there is a problem of governance that we have to discuss how the pricing will be. But most of all, for a lot of varieties, we need also the high-yielding varieties. And how to do that, there's still a lot of sense of work that has to be done. Then I want to say a word because it circulates sometimes in the literature. Instead of transgenesis, some people talk about cisgenesis, that they say we don't use a foreign gene, we take the plant gene itself that we will introduce, so then it cannot be dangerous because it's the same gene. Of course, that is nonsense. There is no difference between a transgene or a cisgene. And on the contrary, at the moment, all the experience show that you're much better off with a transgene because there are still so many regulatory mechanisms at the level of gene expression that we don't know. Very often, if you clone Arabidopsis genes and you reintroduce it in Arabidopsis, then you see no effect. But if you do it in rice, then you see an effect. Because probably Arabidopsis has at the level of the protein modification or some microRNAs that we don't know of, very well built-in systems to make that the gene is not expressed at a high level. And if you want to have a gene expressed at a high level, you better go in a completely different species. And that's work that we still have to learn. So it's not interesting, cisgene, and in concept, if you think that you fool with that regulatory authorities of the society that would accept it more, it's really, there is much more interesting work to do than that. So I could go on like a long time on this science, but I think the discussion with the audience is much more interesting. Thank you.
Thank you. Thank you, Mark. Before I come with a final sentence, is anyone keen to make a further comment? Okay. Yeah, please.
Okay, Professor, but we wouldn't, as Egyptian, third world countries, developing countries, we wouldn't understand more than Germany or more than Europe, more than America, more than Japan. We don't have this science to examine these theories like Germany, like other developed countries. So this is just the point. At when it approved in Germany and in other countries, in developed countries, we can use it in the developing countries.
Yeah.
So please.
Well, you will not see much approved GMOs from Germany, not because of scientific reasons, but because of political reasons. Would you accept GMO crop deregulated in the United States? Okay, then you should have no problem in accepting GMOs because there are many transgenic plants deregulated in the United States. And as you may be more sympathetic with not an example like United States, have a look at Brazil. Brazil is in a nearly comparable situation as Egypt. Brazil has made an enormous progress economically in the last 10 years because they decided to use all scientific and technological possibilities. And Brazil is growing transgenic plants like hell. And Brazil will soon outdate many European countries with the exploitation of the possibility. So I recommend you look at Brazil or Argentina or similar countries and not look at Europe. In the area we have been discussing here, it would be a very big mistake to look at Europe. Klaus?
I just want to say, I was 2 days ago, I was in Cairo at a very nice biotechnology institute, and I was impressed by the knowledge of Egyptian scientists, and you should not put them too low in their quality. I have seen very good scientists there, and I talk here to very good, excellent Egyptian scientists, and they know what they are talking about, and they are absolutely ready to judge what's coming from foreign countries as GMOs. I want to also say that I don't want to give the impression that GMOs are without any problems. There have been mishaps, and the amphora was even mixed up with a Swedish potato, and then it got into trouble. But the amphora potato you mentioned earlier is not for food, it's for starch production, and you should have mentioned that. And anyway, I think the situation, I agree fully with Ingo Potrikos that it's a hysteria, absolute hysteria, and even lawyers are involved in courts. And they, for instance, exempt field destructing by saying, well, that's a minor offense, it doesn't mean anything, although they destroy millions of dollars and
Klaus, may I
euros
cut you short?
Yes,
we
yes.
have more.
And I want just to say the situation is not as simple as it was given by the speaker I criticized, but I can see that there are problems around which should be debated, of course.
Okay. Yeah, there are two more.
Yes. I'm Ahmed El-Khishan. I'm working for Monsanto in the Middle East. And just like to emphasize the information that my colleague says, because we have now here in Egypt is Agiri, Agricultural Genetic Engineer Research Institute, and I'm Egyptian and I got my postdoc from the United States, so we are not less than the scientists outside. And if you mentioned the court of Germany, I would like also, if you could mention the situation of the French court in 2009 that mentioned the decree was not scientific based, and also the report coming from the German Federal Ministry of Education and Research in 2012, and also just 1 week ago, the latest EFSA report on the safety of monitor.
Thank you. Yeah.
Mine is a comment as well. Actually, is a reaction to the commentaries that have been said already. I would like to air out my concern, because this science of GMO has been so much lionized, and the reactions are quite unimpressive. We've had exploitations in areas of physics and chemistry that have gone to the extreme end. Today we have more dangerous things in physics and chemistry around us that we do not even air out serious outright rejections. But when it comes to exploitation of the science of biology, which is still incipient, we give outrageous facets, and it makes the general population get squeamish. The reactions from these uninformed masses are completely deformed. And which future are we having? The resources, water is diminishing. Resources, hunger, people in Somalia are dying. Everywhere people are dying. But if we do not have technologies to have food, where are we going? Okay, right. The traditional methods of breeding can produce. But can't we also support the invasions that are coming probably with a right motive? We do have water shortages. Now water resources are becoming almost war issues. If we do not have these plants that probably can fight against these disasters, what future are we going to have? And this misinformation is completely, I think it is
Thank you for your comments. Thank you. The last comment?
It's a question from an uninformed. I am not a biologist. I come from an electrical engineering background and a sociologist of knowledge. Now, most of your, or some of your data would be in data banks and computer systems. Would some of your new discoveries, or to what extent are your new discoveries, are there potential through bioinformatics, purely through computer systems, instead of going out to the real world, instead of going to vet fair?
So you're talking about plant breeding?
Using computers purely.
Yes. Yes, as I was trying to explain, the quantity and quality of data produced now is such that the approaches to it have to change. And they are changing through the use of databases indeed, and the use of new software tools for data mining, which means that the machines are trained to recognize patterns that we scientists do not recognize without their help. And this whole field of complexity and complexity exploration is probably a new frontier for our science.
Okay, now let me come with my final statement. You don't need to know anything to judge about biosafety of transgenic plants, but to know about the fact that these plants have been used over 25 years on more than 160 million hectares by more than 20 million poor farmers in developing countries without a single documented case of harm to the consumer or to the environment. This is a fantastic safety track record of this technology that everybody should laugh about others who claim that transgenic plants are dangerous. It's so obvious that this is not true. You should not be influenced by what a person who I don't like too much, the old US Minister Rumsfeld said, the old Europe. I'm from old Europe, but I advise you to not listen in this area to voices from old Europe. Try to use this technology with every care to the best of your country and hopefully to the best of the poor people in your country. Thank you.